Hello, welcome to another screencast where we'll be finishing talking about the polymerase chain reaction. We're going to look at some actually applications to this. So the PCR1 screencast dealt with all of this stuff, and now I'm going to try to focus here on this down here, the analysis and interpreting PCR. So the other screencast again dealt with, we'll keep all that, dealt with the procedures of PCR, what was happening over here, some visualization about what was happening in the tubes, and then a little bit about the electrophoresis. And we think we stopped looking here at this slide, the interpretation of this. So let me back up here a second, and I think we had seen in previous screencasts and also in class had talked about how there are areas in the genome that are known to be highly variable, right? So for most of this genome, right, all of these sequences in humans are pretty much the same, and other organisms as well. But we're just happening to look at the human genome here. This is what you guys are sort of most oftentimes interested in. But I just do want to point out that this technique is used for other organisms as well, from bacteria to fungi to plants, etc. So if you look at the human genome as an example, there are areas of the genome that are known to be highly variable, right? And so that is what these things are. So for these other examples here, we're just going to look at chromosome 11. Right? And on this chromosome 11, this locus right here that is called TH01, right? So this is a locus, is known to be highly variable, right, between individuals, right? So if that's known to be true, right, then we can use PCR to sort of detect the differences in individuals, right? So PCR here can be used to help answer forensic type questions. For example, if we have a crime scene and there's DNA left at the crime scene, and we have these 4 suspects that for other reasons have been brought in, right? And we can now know, we can look at the DNA, the highly variable regions, right? This TH01 locus that is highly variable between these 4 individuals, or is known to be, and we can use PCR to amplify the DNA that was left at the crime scene, right? So the DNA at the crime scene has a specific sequence to it at that TH01 locus, right? And what we've done here is we've run this allele ladder in this lane, where we know that there are these variations at that locus, right? So again, alleles are variations at that locus. So we know these. We know that one of those alleles happens to be this long. Happens to be this long, and another allele is this long, and another allele is this long, and so on and so forth. Right? We know that these fragments here are shorter at the bottom, right, and then longer at the top here. So we took a specific primer, and we took the DNA from the crime scene, a very specific primer that was complementary to, lots of primers that were specific for this DNA sequence here. And it turns out that at the crime scene, we were able to amplify, right, a sequence that was complementary to allele number 7. Right? And then this is most likely allele number 3 here. Okay, so the DNA that was at the crime scene, that is not the victim's DNA, it had this allele and this allele present. Right? So were these other alleles are present there? No, because you don't see them amplified. Right? There's no result there. Right? And so the amplification wasn't, the sequence wasn't there, and was not amplified there. But in suspect A, suspect A is showing you that had this allele sequence, right, which is associated with allele 10, right, but did have allele 3. Right. Suspect B here had allele 5 and allele 2. Right. Suspect C has allele 7 and allele 3. Pardon me. And suspect D has allele 3 and allele 2. Now, you may be tempted to say, well, okay, look here, the suspect A has one of those alleles, right, but not the other one. Well, why? It's because everybody's got, these are all diploid organisms, right, so there's 2 copies of chromosome 11, and this has 2 of chromosome 11 and 2 of chromosome 11 and 2 of chromosome 11. So you are you have 2 loci, right? So this suspect A on one chromosome at that locus THO1 had the 10 allele and has the 3 allele. So the genotype of this person is 10-3. The suspect B here, the genotype is 5 and 2, right? So on one they had the 5 allele and they had the 2 allele on the other one. Suspect C here has the 7 allele and has the 3 allele, and suspect D has the 3 allele and the 2 allele. So given all this information, which one of these individuals was most likely, which one of these individuals' DNA is most likely this unknown DNA that was found at the crime scene, which is 7 and 3. So that highly links suspect C here to most likely the DNA at the crime scene. All right. So this is how we can use this here to help answer forensic questions. Forensic questions meaning which suspect was most likely, which suspect's DNA is most likely that at the crime scene. Okay. PCR can be used to help answer paternity or maternity questions. So this is just a graphic of a gel, right? And again, we're looking at these the THO1 alleles here. This is the allele ladder, so we know these, and there are, in this case, we're looking at more different types of alleles, so a 14, a 13, a 12, 11, so on and so forth. So these are all known alleles, and the mother was known to have allele 6 and allele 3 on that chromosome 11, so her genotype would be 6-3. The father's genotype here is 10-5. Child C, that genotype is 10 and 6. Child D is 10-3, and child E is 6-5. Okay. So are these children the offspring of mother and father is sort of the question here. So yes or no, and why or why not? Well, you have to know here that if the father, here's the father, and he's 10 and 5, and here is the mother, and she was 6 and 3. So meiosis, sexual reproduction, the possible combinations here would be 10-6, 10-3, 5-6, and 5-3. These are the potential probable offspring that this parent combination, so if you cross these 2 parents, these are the possible genotypes of the offspring, right? So is this child C potentially a offspring of mother and father? Yes, there's the 10-6 right there. What about this child D, 10-3? Right, well, there we go. There it is right there. That's a possibility. What about this child E, 6-5? Right? Can you get a 6 and a 5? The answer is yes. So right there. Right? So are these children the offspring of this mother and father? Yes, they are. And so we can see this here. So now the question for you would be, what if we had child F, right? And I want you to show me here a PCR result where the child would not be, right? And then let's predict here child G, right? So not let's say either, And the questions of maternity are rarely questioned because the mothers typically know if they had a child or not. So typically we have paternity questions, right? So show me what the results would be. Like the mother is known, but it would be not that father. Okay, so be able to show me what those will look like there. Okay, and then lastly, PCR can be used as a genetic marker or diagnostic tool to answer whether or not potentially you have an allele associated with a disease or a condition or not. Okay, so we've established already that at that TH01 locus that there are several different alleles, right? And those alleles, it turns out, are due to variable number tandem repeats, right? So we have this repeat, and let's go down here a second. So the repeat is this TCAT, right? So the number of times that this TCAT is repeated, right, between this AAA and this CCC right here, right? So each of these hashtags or number values here is the number of times that TCAT has been repeated, so 14, 13, 12. So again, this yields you different fragment sizes, right? So if you amplified, using PCR, if you amplified this specific sequence, right, if you had this allele sequence, you're going to get fragments that were longer. If you had this allele sequence, you get fragments that are a little bit shorter, that are 10 long, etc. And if you electrophoresis these out on a gel, you'd get 14 different fragments there. So if we say, I'm just hypothetically saying here, that some of these alleles are known to be, what we say, linked with or to certain diseases, or a certain disease, meaning that if you have the sequence, if you have this, let's say that you have a 7, right? We would say that then this, having that is a sort of a marker, right, for disease X. Okay. Now, you may have several of these, one or two of these alleles that are associated with disease, and the rest of the alleles are associated with normal or non-disease individuals. So, PCR used as a genetic marker or diagnostic tool, right? And let's say, let's go back a second and say that I pick 7. Oh, let's just pick 6, right here, and 3. So let's say that these two are the markers associated with that disease. Okay, so if we have individual A and we have individual B, and we want to know, the question that we're asking is, do you have the allele sequences, do you have the alleles that are associated with a certain disease, right? So individual A, if individual A has a 6 and a 3, which are associated with that disease, then they would be amplified, and here you see that they are amplified, right? And individual B happened to inherit 10 and 5, and those are not highly associated with that disease, so you would say that individual A has inherited those sequences and individual B has not inherited those sequences. And again, there's 2 of them because you are diploid, right? And so you'd have 2 copies of that chromosome, so the genotype of this person would be 6 and 3, and the genotype of this person is allele 10 and allele 5. Okay, so what about, let's see if you can figure this out here, individual C, right? And what would it look like if you had inherited only one allele, right? Only one allele. So this is, let's say that you are heterozygous for the disease. Okay, so what would your PCR result look like there? Okay, so just a couple of ways here that PCR can be used, right, and interpreted, and why it can be a powerful tool, right? And if you are confused or whatever, bring your questions to class. Also make those predictions. We can talk about those in class. Thanks for listening.